Welcome back. In this last module for Chapter 3, we are going to look at gene expression. Gene expression refers to the body's ability to read the code in the DNA and then translate that into a pattern of amino acids which fold together into a protein. It's really amazing, and it's actually quite a bit like origami. Let's see how. We get our DNA from our parents, right, at conception. So from the moment sperm and egg combine, or at least a few minutes after that, it was determined if you're a boy or a girl, and the color of your eyes and your hair and your skin, and probably a whole bunch of things that we can't see, types of tissue that you have. Anyway, does your DNA change over time? Well, maybe with genetic mutation, exposure to radiation or cigarette smoke, for example. But other than those mutations, does your DNA change? Well, not really. So let's look at this. Here's Carlos. He is an Arctic fox, and in the wintertime he has this beautiful white coat, and in the summertime he has this brown coat. Now think about that. His DNA doesn't change, and yet his physical appearance changes somewhat dramatically. How can we explain this? Well, it turns out that you are born with genes that don't turn on until later in life. And in some cases, genes which turn on and turn off in a cyclical pattern. We see that in humans in menstruation, right? The female sexual cycle is actually an example of genes that are turned off at one point of the month, being activated by hormones, and turning on and doing something at a different part of the month, and then turning off again. How crazy. In Carlos, the Arctic fox's case, we can see here how it works. Along the DNA, there is a region called the promoter region, and this is covered up in the wintertime because of environmental changes, temperature, and maybe the length of the daylight. It causes proteins to cover this promoter region up. And then in the spring, when it starts to grow warmer and the days grow longer, the promoter region is suddenly revealed. Blink, blink, blink. The latent gene is suddenly activated, and it starts to copy that recipe and fold up the little origami protein that is brown, melanin in its many forms, which is exactly what we see here. Can you think of an example in your life? Are you born with genes that don't turn on until later in life, other than those which regulate menstruation? Give two examples. What do you think causes these gene activations? What happens once they activate? Pause the video here and come up with a few ideas. Here are the ones that I can think of. Puberty. Think about this. A 6-year-old boy doesn't make sperm, but does he have the recipe for it? Does he have the gene to produce the proteins and hormones which are going to transform his body through a process called puberty? Yeah, absolutely. What happens once those genes are active? Proteins turn on other genes and turn on mechanisms and cause him to really transform his body. Other examples include aging. This is very strange, but gene expression, or turning genes off and making them latent, has a lot to do with the fact that our bodies really change as we grow older. Baldness, the thinning of our hair or the graying of our hair, that has to do with genetic expression or lack thereof. Okay, so we've established the fact that growth and differentiation can be controlled by gene expression. Differentiation is how a primitive stem cell becomes something more complicated or special, like a red blood cell or a white blood cell. Environmental hormones can activate these genes. This cell cycle is really just one giant example of genetic expression. In order for the cell to do different things, it has to read different parts of the DNA at different times in its life, and we call this the cell cycle. You probably studied this in biology. The first part of a cell's life is called G1, the first gap phase or first growth phase. This is the first opportunity for that brand new baby cell, which was created right here, that midnight hour. That new baby cell is reading its recipe and knowing what to become. A skin cell doesn't read the whole genome. It only reads the part that pertains to being a skin cell. So it makes collagen and it makes keratin. A bone cell reads a different part of the book, and so too do blood cells and egg cells, etc. After growing for a while, the cell pauses to copy all of its DNA end to end, making sure that every letter is exactly the same. And at the end of this phase, there is twice as much DNA as there was before. The cell goes back to work, reading and expressing genes and being its skin cell. And then it divides the genetic material that it had duplicated into two piles through prophase, metaphase, anaphase, and telophase. And then through cytokinesis, it divides all of the fluid and organelles and ultimately divides into two completely separate but genetically identical daughter cells. And each of those cells goes through its own life cycle. The key here is that after that cellular division of mitosis, if everything has worked right, the two daughter cells are genetically identical. Let's make sure we see how that works. Have you studied replication of DNA? DNA replication is duplication, copying the genetic code, and it takes place here during the S phase, prior to mitosis. You can watch this video to really see how it all works. You should understand that. In a second, you should understand that the DNA is made of these G's, A's, T's, and C's, and that the genetic code on one strand is complementary, or the reverse of the genetic code on the other strand. By that I mean, if the genetic code on this part of the double helix says G, A, G, A, we can predict what the genetic code will be on the other strand, the so-called complementary strand, because G's on one strand only match up with C's on the other side. And T's on one strand always and only hydrogen bond or match up with the A's on the other side. You can pause the video right now to double check, but that's what you'll see. Reds and blues and greens and yellows, always on the opposite side. It is this simple rule that allows the DNA to be copied by an enzyme, not shown here, but a complicated machine of a protein jams itself in and splits these two apart. And then other little machines come along and match up the complementary bases to make these base pairs so that the red and blue and the yellow and green are always the same. By the end of this process, we have two complete double helical strands, and they are identical letter for letter. That's replication. Okay. You'll have an opportunity to work through mitosis in lab and watching videos, but we need now to move on to meiosis and to understand how those are, how that process is a bit different. If you're still unsure of the phases of mitosis and what happens during each of those phases, prophase, metaphase, anaphase, telophase, and cytokinesis, you should pause the video here and go back, study that before you move on. Okay, so cell division. Most of our cells multiply for growth and repair through a process called mitosis, where this one cell, called the parent cell, which is a misleading term. We're not talking about sexual reproduction or inheritance. We're just talking about one skin cell that's going to clone itself to create two new ones. The original is called the parent. The identical clones are called the daughters. So when I grow new skin, I don't want it to change. I want it to be the same as it was before. And so each of those cells creates new cells, which create new cells, and they're all exactly the same as they were before. We are doing our best to make sure that we preserve the genetic pattern so that the daughter cells are genetically identical. For growth and repair, it makes sense. If I want to make a baby, though, through sexual reproduction, I want to make cells that are genetically different so that each of my offspring is a little bit different, a little bit more likely to survive, a little bit more likely to have the characteristics or traits that are best suited to the environment. So if you want your offspring to be genetically unique, then your sperm or your egg cells have to be genetically unique. And so we use a similar process, but it's called meiosis, and it actually involves 2 complete cell divisions. So instead of 1 cell becoming 2, we're going to see 1 cell become 2, and then each of those splits so that at the end of this process we have 4. And look at this, they're genetically unique, not identical. This is sort of the beauty of sex, right? There are 7.2 billion people on the planet, and everybody's a little bit different. In the event that that a sperm and egg combine, that fertilized egg is called a zygote. And zygotes are somatic cells, which then go back to using mitosis. Mitosis for a long, long time until they start to produce sperm or egg. And then we use meiosis again. Here's a review of the phases of mitosis. You can pause the video here if you want. Nice graphics showing us the key features of mitosis. During prophase, the chromatin condenses into chromosomes. The spindle fibers begin to form between the centrioles, and the nuclear envelope disintegrates so that the spindle fibers can then grab onto the chromosomes and pull them center stage. That's the key feature of metaphase. In anaphase, the X's are torn apart into V's, so we have 2 rows of V's. And in telophase or telophase, we see the nuclear membrane reformed. For the first time, 2 nuclei inside of 1 cell. The chromosomes decondense into chromatin, and the spindle fibers fade. In cytokinesis, we totally separate the cells by merging or separating the fluid and the organelles. So you get a centromere and you get a centromere. You get fluid, you get fluid. You get mitochondria and so do you. Golgi, Golgi, rough ER. And eventually the cells completely split apart. In meiosis, we want to shuffle the DNA. It is in the best interest of the species, maybe not necessarily the individual, but it's in the best interest of the species to have a lot of genetic diversity. The more diverse we are, the more likely some of us will be to survive if there's a huge change in the environment or some sort of killer virus, right? If we were all the same, then we all have the same vulnerabilities. And so the virus could wipe out everybody, or the environmental change could cause all of us to die. So the beauty of sex is genetic diversity. And in order for sexual reproduction to take advantage of this mechanism, we have to make unique gametes. Gametes are sperm and egg. And listen to these numbers. Just really amazing. Every one of the tens of thousands of eggs produced by a woman in her lifetime are unique. None of them are the same. And did you know that a young and healthy man makes 100 million sperm a day? That's crazy. And believe it or not, every single one of them is unique because of this process of meiosis. So here's the overview. We start just like mitosis with chromosomes that are already duplicated. Remember, these are genetically identical sister chromatids. One of the first significant differences is the first chromosome from the paternal and the first chromosome from the maternal fuse and swap some genetic material through a process called crossing over. so that at the end of the first division, meiosis 1, the chromosomes are already genetically different than what they were before. Then, in meiosis 2, we simply separate those chromatids into individual single-stranded chromosomes. For simplicity, we only see the first pair of chromosomes. And so each of these gametes only has one chromosome. But remember, in reality, there are 23 pairs. And so through that crazy shuffling of 23 different chromosomes, genetic information, we guarantee that every single one of these will be unique. And so a man and a woman, for example, could have many children. And although there may be a certain trait, they might look similar, they might all have the same eye color, most of them, you might be able to tell they're related. None of them is exactly the same unless they're identical twins. Here are all of those little nitty-gritty details. Here we see, let me start over at the beginning. Let me show you here really quick, off the radar. It sounds crazy, but it's true. When you make eggs, ladies, and when you make sperm, gentlemen, you are actually using your parents' DNA. What? Well, here's what I mean. At conception, when the sperm and egg combine, the sperm brought one chromosome to the party, and the egg brought another chromosome. We call this the first chromosomal pair. When your father's sperm and your mother's egg combined, so crazy. He brought one of these and she brought one of those, and he brought one of these and she brought one of those. There are 23 pairs of chromosomes in every cell in your body. During mitosis, we can see them. And you got one from your mom and one from your dad of each of those 23 pairs. These ones are the most interesting, right? The 23rd pair, the egg is capable of only bringing an X chromosome. And the sperm can carry a Y chromosome, but oftentimes carries an X. So you probably know this, but if you get an X and an X, you become a girl. If you get a Y from the sperm combined with the X from the egg, you become a boy. Okay, so keeping this in mind, when we go to this PowerPoint, what we actually see here is mom's duplicated first chromosome. Just like in mitosis, there is a DNA replication phase prior to meiosis. And so this red X is actually the already duplicated maternal first chromosome. This blue one then is dad's duplicated first chromosome. And like partners finding each other on the dance floor, the first chromosomes form what we call a homologous pair. The first paternal chromosome would never hook up with the second maternal chromosome. It would be as scandalous as me dancing with your, I was saying with your wife, with your husband. Boy, that would be really scandalous, right? Here's what I mean. If we all brought our partners to class and we had a dance party, you would very easily find your partner on the dance floor, wouldn't you? You wouldn't confuse your partner for my partner. And so in the same way, these homologous pairs are chromosome 1 and 1 and chromosome 2 and 2. They don't have any problem finding each other. And so they swap a small portion of their genetic material through some serious dirty dancing, and what we're left with are these hybridized chromosomes. Now, genetically unique. That's the most important part of prophase 1. In metaphase 1, all the chromosomes line up in the middle, just like we had seen in mitosis, but here's the deal. They're already genetically different. And so it actually matters which side of the line they end up on. In this scenario, the big blue and the little red are on top. But it could just as easily have ended up that the little blue and the big blue were on top, and that will actually matter. This process is called random assortment or independent alignment. It happens during metaphase 1. and it is the second of two mechanisms that guarantees that the gamete will be genetically unique. Crossing over, which happens in prophase 1, and independent assortment or random alignment, which happens in metaphase 1. Everything else is really just like mitosis. At the end of meiosis 1, we have 2 cells. And at the end of meiosis 2, we have 4 cells. And here's the key. They're haploid. Instead of 23 pairs of chromosomes, they have 23 single chromosomes. And so the sperm can combine with the egg to give us that fertilized egg, which has 23 pairs, one from mom and one from dad. Here is some great ideas for you to really make sure you know this information. If you just read about it, you can trick yourself into thinking you really understand it. But anytime you put something down on the table and physically work with it, either by writing or drawing or sculpting or talking or singing or rapping, then it really becomes clear what you know and what you don't. So I recommend trying some of these ideas. I'm going to do one more video when we come back to walk you through transcription and translation and gene expression.